Our esteemed guest has arrived. We will be starting very soon.
So, good morning.
Good morning, sir.
Good morning. Good morning.
So, principal sir is just joining. So,
No
we'll start.
problem. No problem. I'm just wait.
Sir, shall we start?
Yes, please start.
Thank you so much, sir.
Good morning, Dr. Shiv Kumar.
Namaskar, sir. Namaskar. I'm sorry, sir. I'm traveling.
Namaskar.
Good morning, sir. Please start.
Okay, sir. Okay, sir. A very good morning to everyone. I am Dr. Richa Arora, working as an assistant professor in the Department of Chemistry of Shivaji College. A warm and hearty welcome to one and all to a talk on chemistry, a central science of nature. This event is being organized by the Department of Chemistry, Shivaji College, under the aegis of DBT-sponsored Star College scheme. This event is being broadcast live on the YouTube channel of Shivaji College, whose link is given in the chat box. Let us all seek blessings of the ultimate power above us and join hands in prayer.
Shubham kurutvam kalyanam arogyam dhanasampadah. Shubham kurutvam kalyanam arogyam dhanasampadah. Shatru buddhi vinashaya dipajyotirnamostute. Dipajyotirnamostute. Shubham kurutvam kalyanam arogyam dhanasampadah. Shubham kurutvam kalyanam arogyam dhanasampadah.
First of all, I would like to thank and welcome Professor Ashok Kumar Prasad, our chief guest and esteemed speaker of the program. Professor Prasad is currently Dean, Faculty of Sciences, Head, Department of Chemistry, and Intellectual Property Rights Chair of University of Delhi. On behalf of the Department of Chemistry, I would like to present a virtual bouquet of flowers as a token of our appreciation for Sir. Thank you so much, sir.
Thank you.
I extend my gratitude to Professor Prasad, who, despite having so many commitments, promptly accepted our invitation and is here with us to share his vast knowledge and experience with us all. Thank you so much, sir. We indeed are very lucky that we are going to learn chemistry from such a renowned personality. I welcome Principal of Shivaji College, Professor Shiv Kumar Sede. Sir has been a motivating force behind this event, helping and guiding us throughout. Sir, I humbly request you to take over the virtual stage to formally inaugurate today's event. Please, sir.
Thank you very much. It's my proud privilege to welcome our Honorable Chief Guest, Professor Ashok Kumar Prasad ji. Professor Prasad is like a family for Shivaji College, and he is very closely associated with our college. And sir, I really welcome you once again to your own college. And a very warm good morning to all of you, my dear colleagues and dear students. I'm very happy that Department of Chemistry has organized this webinar on very interesting topic. I'm sure that you will enjoy and you will get benefited from the talk of Professor Prasad. Thank you very much, sir, for sparing time for my faculty and students. Thanks a lot, sir. Thank you very much.
Thank you.
Thanks a lot, sir. I always fell short of words when I have to express gratitude towards you, sir. Thanks a lot for being there at every turning point. Now, I would like to introduce our coordinator of DBT-sponsored Star College Scheme of Shivaji College, Professor Darshan Malik. Her constant support and dedication always make us push further and excel at everything we try. Ma'am is not here with us due to prior engagements, but she had sent her good wishes, and especially for Professor Prasad. Thanks a lot, ma'am. Now, let me introduce you with the protocol of the webinar. All the participants are muted on purpose so as to avoid unnecessary background noises, which might distract you from listening to the speaker clearly. But you have a golden chance of interacting with our speaker. You can submit your questions or queries and comments in the chat box of Zoom anytime during the talk. Your questions will be addressed after the talk in question answer session. I know you all must be eager to hear Professor Prasad, so without any further delay, I would like to introduce our guest speaker in the form of a short summary of his endless achievements. Professor A.K. Prasad did his PhD from University of Delhi in 1990 in the area of synthesis of bioactive polyphenolic natural products under the kind supervision of Professor A.C. Jain. After spending about a decade as postdoctoral fellow and visiting scientist at the University of Southern Denmark, University of Copenhagen, Max Planck Institute for Molecular Physiology, Germany, La Spezia University, Rome, Italy, and University of Massachusetts Lowell, USA, Professor Prasad joined the Department of Chemistry as reader in September 2001 and subsequently became professor in June 2009. Professor Prasad is a member of several national and international professional societies and has published over 200 research papers in journals of international repute, along with 10 patents. He has presented more than 175 research papers at different national and international conferences and symposia, and delivered 75 invited lectures in the universities, institutes, and industries in India, Australia, USA, Denmark, Germany, Italy, Holland, Switzerland, Japan, China, etc. Presently, Professor Prasad has a research group of 8 PhD students and 5 postdoctoral fellows. His research interest lies in the area of synthesis of sugar PEG-based amphiphils as novel carrier agents, modified nucleoside monomers for therapeutic oligonucleotide synthesis, biotransformations, natural products chemistry, and synthesis of bioactive heterocyclic compounds. He is handling various research projects, including bilateral projects funded by DRDO, DBT, DST, NMPB, and University of Delhi. He has been a recipient of visiting professorship at JACE, Japan, 2015 to date, ACCTI Excellence in Carbohydrate Research Award 2015, ISCB Award for Excellence in Chemical Sciences 2014, ETCR Best Paper Award 2012, visiting Associate Professorship at SDU Denmark 2009 to 10, CRSI Young Scientist Award 2007, DA and IDA Denmark Fellow 1992 to 96, National Scholarship Award, to name a few. He has been the editor and guest editor of Biochemy, published by Elsevier, Indian Journal of Chemistry, and Trends in Carbohydrate Research. He has also been involved in the organization of many international and national conferences and symposia in the department. So, I will now hand over the virtual stage to you so that you can share your knowledge on the topic, chemistry, a central science of nature. Over to you, sir.
Thank you. Thank you very much for your kind introduction.
You're welcome, sir. The patient.
So, can you see the slide?
Yes, sir. Yes, sir.
So, thank you very much to Shivaji College, where I have served as a different capacity in governing body for quite a few years. And it is a pleasure. I always enjoy visiting Shivaji College. And I have seen the transformation of Shivaji College for good, for so many years. And I would like to take this occasion to thank my friend and principal of the college, Dr. Sukumarji, for taking the legacy of Shivaji College forward, and then Dr. Darshan Malik, for helping to organize this webinar, along with all chemistry faculties, particularly Dr. Arora and Dr. Sahu, for inviting me to give this talk in the college. So it's a pleasure speaking in Shivaji College and for the students of Shivaji College. So, today I have chosen the topic, chemistry, a central science of nature. We don't have to tell when the science started. In fact, nature has started science billions of years ago, and started making compounds which forms the basis of life later on. If we go back and see, there is one theory that how life arrived on the earth. And one of the theories out of many theories is primordial soup theory. And according to this, the living system evolved on this earth, first from the aqueous solutions of organic compounds. So this aqueous solution of organic compounds, almost 4 billion years ago forms the basis of life. How this aqueous solution of organic compounds was formed? This theory says that this aqueous solution of organic compounds formed, it is accumulated in primitive water bodies, of the earth as a result of endogenous abiotic synthesis. Even we observe today that if you leave something in water for a long time, water becomes contaminated and then life appears in this. But we are not talking about today. We are talking about billions of years ago when there was no sign of life. And then this theory says that because of abiotic synthesis in water bodies in those days, and this could happen due to due to natural phenomena, due to the light energy coming from the sun, due to extraterrestrial delivery of extraterrestrial delivery by cometic and meteoritic collisions, which led to the formation of this primordial soup, which contains many organic compounds. So if you can see, this theory says that in primitive water bodies, because of the sunlight, because of the collision of different terrestrial bodies, slowly synthesis of a small organic compounds started in water bodies. We know that oxygen was there in the atmosphere, carbon was there, nitrogen was there in the atmosphere. So all these slowly in millions and billions of years, small molecules started coming because of carbon and oxygen first, then carbon and hydrogen, and then carbon, hydrogen, nitrogen, and then slowly amino acid kind of molecules get synthesized. And these amino acids ultimately led to the synthesis of proteins. And then from proteins, slowly in the other pathway, these sugars came, and these nucleobases came, DNA appeared, and this is how life evolved 400 billion years ago. If you will try to imagine what this 4 billion years number is, 1 million year is 10 lakh years. So 10 lakh years is 1 million year, and 100 million is 1 billion. So you can imagine we are talking about long ago, when this primordial shape, according to this primordial shape theory, life originated at that time. And there also chemistry involved, because it started synthesizing a small molecules due to the different kinds of energy which was coming, and the nitrogen present in the atmosphere led to the synthesis of a small carbon hydrogen molecules, carbon hydrogen oxygen molecules. Then it says that the life began on Earth at least 3.5 to 4 billion years ago, and it has been evolved ever since. It is evolving. Evolution of life has not stopped. I think the most common and the most recent example is that every day we see in the newspaper that it is, coronavirus, alpha, beta, gamma, delta, different mutants of coronavirus is appearing, and which is leading to all the complexities in controlling this disease. So even coronavirus, which is not a well-organized cell, it is RNA-based organism. Even this virus is evolving every day. It is getting mutated to new virus of the same family. So this evolution has not stopped even today, ever since it is evolving, since this life appeared almost 3.5 to 4 billion years ago. And if we try to see, this is all from Google. I have not made it. I have not done anything. This is just for the information I have collected it. So the first prokaryotic cells came into the picture when the life evolved. When we say prokaryotic cells, it is a very primitive cell. Even there is no nuclear membrane. So some nuclear material is there in the cell. It still exists. And then, in the cytoplasm, the complex organelles which is there in the present cell is still not there. So even it's a very primitive cells which appear first. And then it says slowly photosynthesis started, and this photosynthesis accumulated oxygen in the atmosphere. And then the complex cell, eukaryotic cells, came into the existence almost about 2 billion years ago. And then came bacteria, protozoa, and this is how life evolved, and ultimately Homo sapiens appeared. based on the study of skulls, that has been found in different regions of the world, the analysis of these skulls says that these Homo sapiens or the modern humans began spreading across Eurasia, Europe and Asia, from Africa and adjacent Southwest Asia about 50 to 55,000 years ago. So we can assume appearance of these Homo sapiens, some maybe 90,000 years ago or 1 lakh years ago, and they started spreading in different parts of the globe, 55 to 50,000 years ago. So this is how life evolved, and now we see that how complex the life is. And now from unicellular to multicellular and multicellular to multiple tissues and complex organism has evolved. That's the human sapien, Homo sapiens, and we the human beings, with a very complex kind of science going on inside the body all the time. I will try to discuss this Nobel Prize, which was given in the area of physiology and medicine in 2015. So this Nobel Prize was given to this lady from China, Yu Tutu, and other two scientists, Omura and Cantwell. Omura and Cantwell got 50% of the Nobel Prize of 2015 in the area of physiology and medicine for his discovery concerning novel therapy against infections caused by roundworm parasites. And the other half of the prize went to Yu Tutu, and to her, this prize was gone for discovering a novel therapy against malaria. So you see the disease. In 2015, the Nobel Prize for Medicine and Physiology, it went to scientists who discovered compounds from nature for the treatment of parasitic and roundworm diseases and for the treatment of malaria. What is this anti-malarial compound? This anti-malarial compound is this, which is called, artemisinin. So this artemisinin was isolated from a plant called Artemisia annua. And it was isolated by this UU2. And, after isolating this compound, she led to the development of this compound as anti-malarial drug. If you find a compound from nature, you do not know what use of this compound is. So to find out the use of that compound, you have to test for 100 different diseases. And once the compound shows some activity for some disease, then you have to do further testing, animal trial, all these phase 1, phase 2, phase 3 trials before the compound comes as a drug in the market. So it's a very long pathway. We know that the vaccine has come for coronavirus or COVID-19. But so far, no drug has come because development of drug takes long time. It has to be tested at many different levels. So whatever drug, was given last time, you would have heard one of the drug was ivermectin. This ivermectin is nothing but this compound, which was isolated by Omura and Campbell for roundworm and for the treatment of roundworm and parasitic diseases. So this is anti-parasitic compound. This was also used for the treatment of COVID-19. So this shows that chemistry doesn't found itself in the boundary of synthesis or in the boundary of study of different chemical compounds. Rather, it breaks its boundary and it enters into the medicine and biology. And this is a good example of this. Now let us go to the same year in 2015, the Nobel Prize which was given for chemistry. And this Nobel Prize in chemistry in 2015 was awarded jointly to Thomas Lindahl and Paul Modric. And the other scientist was Aziz Sankar. So these three scientists got the Nobel Prize of Chemistry in 2015. And why they were awarded this Nobel Prize? They were awarded for mechanistic studies of DNA repair. We have just now seen that this Nobel Prize in 2015 awarded to physiology and medicine for discovering these compounds as a drug. And the same year, in chemistry, the Nobel Prize was awarded, for the work as a mechanistic studies of DNA repair. So this is the DNA, and DNA is always damaged, as always, get some change due to different reasons. If you want to maintain, the genetic information intact, it has to be repaired constantly. So there is a natural process of repairing of DNA. If this process fails, then you can say that sky will fall, because even one change of one base pair, I don't know how many of you know, I will tell you in a moment, it led to the disease which is called sickle cell anemia. One base pair in DNA, billions of base pairs are there. Even out of those 1 billion or 2 billion or 3 billion base pairs, even one is, it causes serious disease. So you know how important it is to repair DNA. And therefore, this Nobel Prize was given in 2015. Why I showed you these two slides is to impress upon you that, chemistry is really a central science of nature. Either it is DNA repair, either it is synthesis, either it is synthetic biology, or either it is drug development. Everywhere chemistry is there. Either in our food, in our clothing, in our shelter, chemistry is involved everywhere. So it's a really central science of nature. This is the prize which was given last year. This year Nobel Prize has not yet come. And this prize was given to this lady, very important, because the first time two women shared the Nobel Prize. Emanuela Carpentier, she is from Max Planck Institute of Science and in Berlin, and Jennifer Donda from University of California, Berkeley. And this Nobel Prize was given to them for development of a technology. And this technology is used for gene editing. Editing means you can edit. You can change, as per your design. So this is called editing. So you can edit the gene. And this technology developed by them is called CRISPR technology. What this CRISPR technology is? This CRISPR technology, it's a short form. It is called clustered, regularly interspaced, short palindromic repeats. So this is CRISPR. What is the role of this CRISPR technology? We know that DNA is a very important molecule in every cell, and information regarding that particular organism is all there in DNA. DNA has different genes, and these genes are responsible for different properties of a particular organism. And how these properties is guided by synthesis of different proteins, from the information coming from this genetic region of DNA. So if this genetic region of DNA has information for a black hair of a person, it will synthesize a particular protein, and that protein will only make black hair. If you go to Europe, their genetic material, which is for the color of hair, may be different, and then it will synthesize a different protein, which will lead to the synthesis of a brown hair. It can have a different protein which can lead to the synthesis of a curly hair. So this protein, which changes all these properties, it is synthesized based on the information in the genetic region of DNA. Similarly, you take, the black eyes or the brown eyes, the information is there in a DNA, and this information has to be intact. If this information is changed, then things will change. And therefore, gene edition is a very important technique, and therefore Nobel Prize was given. What can you do with the gene editing? if you know that if some genetic region is mutated and it is synthesizing wrong protein which is causing disease, you can disrupt this gene by inserting a different gene here. So now this gene will no more be there. So it is called gene disruption. You can do gene correction. So if you know that this gene has mutated here, you can take the right gene and introduce the right gene in the DNA. So this is called gene correction. You can do gene insertion. If you want to enter an entirely different gene in the DNA, so you can synthesize that duplex and you can introduce in the DNA, and then you will get an entirely different gene, which will do some new thing. And therefore now we are well aware that this different variety of genetically modified plants or different things are coming. So this CRISPR technology is very important, which was developed last year and developed in the last couple of years, and for which the Nobel Prize was awarded to Carpentier and to Donda in 2020. From all this, you see that study of this genetic material, DNA, is very important because it have all the information. So if there is a change here, it can have a big consequences on the organism, on the particular organism, cause many different diseases, or like we know that when the genetic material of this coronavirus is changed, it becomes alpha to beta, beta to gamma, gamma to delta, and it may lead to some modification which can be very dangerous. So all the drugs which is used today do not work on that mutated variety. So therefore, it is very important to study this CRISPR technology and use it properly. And it is very important to study the DNA. So, with this, I would like to tell a little bit about the DNA. So DNA is nothing but a double helix. It's a double helix. And one of the important properties of DNA is DNA replication. So this double helix, it can replicate to the identical to DNA. How it replicates? First it gets bifurcated from the double helix. So one end, it starts separating from one helix to the other helix. And then both these strands, for example, this blue is the original strand. So both strand works as a template. On this template, when they separated, they have adenine, cytosine, thymine, guanine, adenine. These bases are there. And we know that adenine always attracts thymine. So, this adenine attracts thymine, cytosine attracts guanine on this platform, this thymine attracts adenine, and then, other strand gets started synthesized here. But this synthesis is guided by the base sequences on the original strand. So therefore, these two strands of original DNA ultimately lead to the formation of two separate DNA. So this process is called replication, and therefore I always say that DNA is like for chemists, it's a molecule. Otherwise, this molecule can form two identical DNA. So this is a kind of a molecule which is on the boundary of living and non-living. So you can making two daughter DNA from one DNA. So this process is called replication. The other very important process is called transcription. And what is this transcription? So this is DNA double helix. And DNA has the genetic region where all the information is there. And this genetic region is because of the duplex. Now, this information here, when it is read by a protein, that protein synthesizes messenger RNA. So what happens? Where the information is, that portion gets separated. So two strands are separated. And then you can imagine that each strand has different base sequences. This will have this base sequence. So one strand is called information strand. So this is the information strand, and this is called, the another strand, complementary strand. And on this complementary strand, these bases attract corresponding bases. So if it is a guanine, it attracts cytosine. If it is adenine, it attracts uracil instead of thymine because it is RNA. And thus, a new strand gets synthesized, which is called messenger RNA. And all these bases which comes here, it is same base which is here. So if it is G here, C will come here. You see it is C. If it is A here, U will come here, and you can see it is T. So in DNA it is thymine, and in RNA it is uracil. So this is the only change. Otherwise, same basis comes here. And when this messenger RNA is synthesized, it comes out of the nucleus. It takes the information from DNA, from the genetic region, it comes out from the nucleus and it goes to a cytoplasm. When it goes to the cytoplasm, it is read by different mechanism, different machinery, and then 3 bases code for one protein. For example, if it is GAG, for example, GAG here, it codes for glutamic acid. If it is CGA, for example, it is CGA, it will code for arginine. So these 3 bases will read, and a guanine glutamic acid will come. Next 3 bases will read, and then arginine will come, and arginine and glutamic acid will form a amide bond. So, and then slowly a polymer will synthesize, and this led to the synthesis of protein. So this process from DNA to messenger RNA is called transcription, and from messenger RNA to protein, it is called translation. This is called transcription because in this case, the language is same. DNA also has sugars and bases, and messenger RNA also has sugars and bases, because it is the same language, and therefore this process is called transcription. However, once the messenger RNA is synthesized, then it leads to the synthesis of protein, where the units of protein is not the nucleoside. Unit of protein is amino acids. So now the language has changed from nucleoside to amino acids, and therefore it is called translation. And this whole process from DNA to RNA to protein, it is called central dogma of molecular biology. So this is the basis of whole molecular biology, that how this whole process takes and proteins are synthesized. This I wanted to tell you just to understand that how important, the DNA is there, which synthesized billions of years ago and make the basis of life. So, DNA is nothing but a double helix. It looks like this, but if you closely see with the eyes of a chemist, it looks like this. So here is a sugar, a pentose sugar is there, 2-deoxyderibose, and here is the bases, cytosine, guanine, adenine, and thymine. So these bases are here, and outside this phosphate backbone is there. You can see that 2 nucleosides are attached by phosphodiester bond with a phosphoric acid. Third hydroxyl of phosphoric acid is free, and then because it is acid, it is O-. So here is the bases. These bases have the information. 4 letters are there: cytosine, thymine, adenine, and guanine. Out of these 4 letters, you can write anything on the bases, and those writings are taken as information. So they are signature of gene. And therefore, these bases are very important. And this, cytosine is always complementary to guanine. Thymine is always complementary to adenine. So this is how one oligonucleotide, forms duplex with another oligonucleotide. But it cannot be any oligonucleotide. In the next oligonucleotide, the base sequences should be fixed. For example, if the first one has cytosine here, thymine here, and guanine here, the next oligonucleotide will have just the opposite bases. If it is cytosine, then it will have guanine. If it is thymine, it will have adenine. If it is thymine, it will have adenine. And if it is guanine, it will have cytosine. So only the complementary base can come and form a duplex. Any base cannot come and form a duplex. So there should be 2 complementary oligonucleotides to form a DNA. And these 2 complementary oligonucleotides, if you see, oxygen is up here, the oxygen is down. So it always goes in an anti-parallel way. So two oligonucleotides in an anti-parallel way, and these two oligonucleotides have to be complementary to form the DNA. And this DNA is very important molecule because all the information are written here. And a lot of negative charge here on the both sides. Why this negative charge is there? Nature is very smart. Nature thought that make the DNA such a robust that no nucleophile can enter inside. If nucleophile will be enter inside, then they will change these bases. And if the bases are changed, this is gene mutation will take place and it will lead to a disease. And therefore, all this minus charge is there on the backbone on both sides. And this minus charge doesn't allow the nucleophile to enter here because here is the information zone. So no nucleophile can enter into information zone and damage the DNA. Even with this protection, DNA mutations takes place, leading to particular disease in some people, or there is a natural process of DNA repair if this damage takes place. So, I was telling you that this is a sickle cell anemia disease. Normal blood cells in the blood vessels looks like this, like rose petals, very soft, and it flows very easily. But in sickle cell anemia, it becomes hard, like, a semel flower, and it becomes hard, elongated, and therefore it doesn't flow very easily. These red blood cells is red because of iron here. And hemoglobin has a iron center. This change of structure of hemoglobin, makes it elongated, and therefore its iron carrying capacity is also decreases. So sickle cell anemia, because it doesn't flow in the blood, it takes very less iron than the normal red blood cells, and therefore it causes severe pain in the body and weakness, and it can lead to death. And do you know why it happens? It happens because in this genetic region, which led to the synthesis of hemoglobin protein from messenger RNA, one of the bases changes. What base? Sickle cell anemia is caused by change in a single base of DNA. What should be the original? Originally should be GAG triplet in the gene that for the beta subunit of hemoglobin. So this GAG changed to GTG. So this A is changed to T. Somewhere in the genetic region which leads to the synthesis of hemoglobin protein, A is changed to T. Adenine is changed to thymine. Now see what change is done in the hemoglobin protein. When this GAG code for an acid called glutamic acid, these amino acids. These amino acids enter into the protein. If it is GTG, it codes for valine. So valine enters into the protein. So hemoglobin, when this modification takes place, it becomes GTG somewhere in the genetic region which lead to the synthesis of hemoglobin. All the glutamic acid is replaced by valine in the hemoglobin protein. Now try to see, glutamic acid has a extra carboxylic acid, and this forms, the backbone of amino acid proteins. So this carboxylic acid makes the whole hemoglobin highly polar. When it is changed to valine, instead of carboxylic acid, it is isopropyl group. So it makes the whole hemoglobin protein very non-polar. When it becomes non-polar, it changes shape from this to this, because now it is not water soluble, and therefore it gets elongated. Earlier, it has a very good interaction with the water. Now it has less interaction, so it becomes elongated, and then its iron binding capacity also decreases, so it carries less oxygen, and therefore this disease causes. So therefore, I told you that how important this DNA, that even change of one base pair can lead to a dangerous disease, and therefore, 2020 Nobel Prize or 2015 Nobel Prize for gene repair mechanism, is justified. What we do in the lab is that we synthesize different modified nucleosides or different modified sugars, and we use lipase for synthesis of different nucleosides. Why we chose sugar? We chose simple glucose, glucosamine, for synthesis of these compounds, modified sugars, and this sugar where two primary hydroxyl group is there, two secondary hydroxyl group is there. This is pyranose sugar, again two primary hydroxyl group is there, and three secondary hydroxyl group is there. We synthesize all these compounds for synthesis of modified nucleosides. Here are some of the papers which we have published in the area of synthesis of modified nucleosides. So all this is modified nucleosides we have synthesized, using this sugar and the lipase enzyme, and this sugar can be derived from glucose. And we have led to the synthesis of this kind of dinucleotide or polynucleotide, and we have led to the synthesis of this kind of amphiphiles, using this pentose sugar where two primary hydroxyl group is there, or furanose sugar where two primary hydroxyl groups is there. And this kind of polymers is a amphiphilic polymer, and therefore it can form micelles, and we have tried to encapsulate this drug into the micelle. I will tell you in a moment. We have also tried to synthesize these kinds of macromolecules, which looks like crown ether, and it has crown ether properties. So these are the different types of compounds we have synthesized using sugars. I will not spend much time on this synthesis. I will just show, some structures and the compounds. So we have synthesized many different modified nucleosides, using very simple sugars here and the lipase, and all this is published in different papers. And we have confirmed their structures by X-ray crystallography. I will not go in detail of the synthesis, but this is all published work, and if anybody is interested can see this. So this is the DNA, how it is synthesized, one oligonucleotide, and it comes one by one and it forms this 5-carbon oligonucleotide, 5 nucleoside oligonucleotide, 6, 7, 8, 9, 10. So this is how it can get on added. And then these oligonucleotides you can also show like this. So this is the backbone, this backbone is this, and all these bases, because these bases are different, these bases are shown by this sticks here. So this is the backbone, and this backbone is nothing but this sugar and phosphate, sugar and phosphate, sugar and phosphate, sugar and phosphate. So this forms the backbone of oligonucleotide, and these are the different bases here. So if you have modified nucleosides, you can synthesize oligonucleotide like this, but here the bases will be same. However, the sugars here on this backbone will be modified, and we have all synthesized modified nucleosides which can lead to the synthesis of modified oligonucleotide. Now let us see what is the use of these modified oligonucleotides. Just now I told you, central biological dogma where DNA synthesizes messenger RNA and messenger RNA synthesize proteins. Now let us say if some mutation has taken place in DNA. So if that mutation has taken place, it will lead to the synthesis of wrong messenger RNA, because messenger RNA is synthesized based on the information there in the DNA. And if it is wrong information here because of mutation, wrong messenger RNA will be synthesized, and which will lead to the synthesis of wrong protein. And if wrong protein is synthesized, then it will cause one particular type of disease. Just now we have seen in the case of hemoglobin, in sickle cell anemia. So one base change here led to the synthesis of a different triplet, and this different triplet led to the synthesis of hemoglobin, which was rich in a particular non-polar amino acids than glutamic acid, and that led to sickle cell anemia. How to stop this? One can do this. One can synthesize a oligonucleotide with a modified sugars here, but completely bases. If it is T, one can put here A, and if it is C, one can put here G, so that it can form hydrogen bonding, and so on and so forth. And then if you put in the cell, what will happen? It will cover all this information which is there on the messenger RNA. And once the information is covered, protein synthesis will stop. So if some wrong messenger RNA is there, you can stop its expression. And here is the use of this modified oligonucleotides, which we have synthesized. and a lot of such modified oligonucleotides have now entered in the market as a drug. So this technique is already used for therapy of different kinds of diseases. In the next portion of my talk, I have synthesized. I told you that we have synthesized polymers where there is a sugars, and these sugars are here, and then here is a polyethylene glycol. So sugar and polyethylene glycol, and we have synthesized. We have done the transesterification reaction in presence of lipase, and this OH and this ester. This OH replaces the methoxy group, and it forms a new ester bond, and this OH replaces another PEG 600 dimethyl ester group and forms, and thus it PEG sugar, like this structure. Polyethylene glycol is highly water soluble, and these sugars, you can see here, all the hydroxyl groups are protected. So this is highly nonpolar. And when we put this, drop this in water, it forms micelles. Water soluble portion goes out and insoluble portion goes in. So all the sugars goes inside because they are nonpolar, and because it is in water, and all water soluble portions, which is polyethylene glycol, it goes towards water and forms a micelle-like structure. And here you can place a nonpolar drug which has very less bioavailability, and then you can deliver it to the right target. That's what we have done. So these are some of the papers we have published in the area of amphiphilic polymer synthesis and their application. And the first application was that we encapsulated this drug inside the micelles we have synthesized. This is a drug which can be used for photothermal therapy and photodynamic therapy of cancer. So if this drug can be taken into the cancerous tissue, and if you irradiate with the near infrared light, a small amount of heat is generated, and this heat is enough to kill the cancerous tissue, and therefore it is used as photothermal therapy. It is also used in photodynamic therapy, because when it is irradiated with the near infrared light, it converted molecular oxygen to the singlet oxygen, and singlet oxygen is poisonous. So this singlet oxygen can kill the tissues. So if you can bring this compound in the tumor and irradiate it with the near infrared light, it can kill the cells of the cancerous tissues by two ways, by photothermal therapy and photodynamic therapy. And therefore, but one drawback is for this compound. This compound is very less stable in aqueous system. You see, in 5 days, it almost disintegrates to zero. So its stability has to be increased. And we discovered that when it is encapsulated, its stability increases manifold. You can see here that when it is encapsulated in the micelles of these two polymers, where the percentage of loading is very high, 6.5% and 15.4%, you can load here, so it will go inside. So it will be there in the environment of non-polar environment. There is no water, because outside the water is there. So it is not exposed to water, and therefore its stability increases manifold. You can see here that at 25 degrees centigrade, even on 12 days, it is almost intact, no disintegration. Even at 37 degrees centigrade, you can see, on 12 days, very less degradation is there. It's 85%. So, and this blue is the micelles, which has come from polymer 3, and this is polymer 3. So the micelles derived from this polymer. If you encapsulate this endocyanin grain, the disintegration is very less. We have also shown here that these micelles can encapsulate compound, and it can enter into the cell. That is called cell penetration. So this micelle can penetrate into the cell. So what we did is that we encapsulated Nile red, which is a red color. When you irradiate with the UV light, it becomes red. So we encapsulated Nile red, and then we incubated cells with this micelles, and we saw that these micelles enter inside the cell, because all these cells are now red. And this is the blue color is the nucleus because it is colored with the DAPI, which if you color the nucleus with the DAPI, it will shine blue in the UV light, and the cells are shining red because they have Nile red in this. So we proved that these micelles can carry the cargo inside the cell. This is very important. The small molecules. This is the normal tissue, which is very tightly packed, and this is cancerous tissue, which is the cells very loosely packed. Because in cancerous tissues, a lot of cargo has to come because cells are dividing every second. And when cell divide, it requires all those proteins, all those amino acids, because the daughter cell, two daughter cells should be identical. So all those compounds are required, and therefore a lot of cargo is delivered all the time because the cells are dividing. However, normal tissue doesn't divide so fast, and therefore very tightly packed here. If you will give a small molecule, for example, the drug structure I have shown, it will go here also and it will go here also. And therefore, all of you know that, chemotherapy has a very strong side effect, and, patients almost, get different disease because after every cycle of chemotherapy. Because of this, because these chemotherapy drugs, they enters in the normal tissue also, in the cancerous tissue also. However, if you encapsulate it, then the size becomes bigger, few nanometer. And therefore, it doesn't enter here freely. However, it can enter in the cancerous tissue freely because cells, there is an intercellular space, and then it can enter fast. And when it enters here, you know that our micelle is made of ester bond. So ester bond is hydrolyzed by lipase, and then drug can be released in the cancerous tissue, not in the normal tissue. And then, this is called selective delivery of drug. So therefore, this encapsulating drugs in the micelles and then delivering it at the target, is a very important area, and many people are studying it. This is the last part of my talk, where these three scientists, they got the Nobel Prize, I think in 2020, and then 20 or 2019, it is hiding here. And their Nobel Prize was given to them for design and synthesis of molecular machines. So they synthesize many kinds of molecules which really works as a machine. And one of the examples is that they synthesize a molecule like this, and if you put this molecule on the surface, particular surface, it starts moving. So it behaves like a machine. And for this work, they got the Nobel Prize. And Stardot is a very, very famous scientist, who has contributed a lot in this area. What we have done is that we have synthesized crown ether, and this is the crown ether structure, simple crown ether, 24 crown A. And this is the molecules we have synthesized. It also looks like crown ether, but it has one of the sugar molecule inside it. So this sugar we synthesized from glucose, and we have introduced it into the crown ether, and that is very important. Why we have done this? Because this crown ether is used very much, for synthesis of molecules like this. If you see this crown ether, all these oxygens are inside, and this oxygen has lone pair of electrons, and these lone pair of electrons, it can attract positively charged molecules. So if you have some positively charged molecule, it will go inside and sit here because of its attraction with this crown ether. And you can synthesize molecules like this for pseudoreactants. You can synthesize molecules like this. When both the ends are capped, it cannot come out. Here it can come out, but it cannot come out. And the such molecules are called rotagens. Or you can synthesize catenins where there is a ring. So the two rings are one inside the other. So these types of molecules have been synthesized, and they have been used to prepare molecular machines. We have synthesized this type of crown ethers, and because of this ring here, this crown ether is less flexible than this, and because of these chiral centers, chirality has also been introduced in the crown ether. And then we have synthesized dibenzyl ammonium salt. So this nitrogen is positively charged, and then we have demonstrated that this can enter into this crown ether, and this positive charge interacts with the lone pair of electrons in all these oxygen and nitrogens. So this is what we've done. So we have synthesized molecules like this, which is a molecular machine. And we have also demonstrated that, until unless there is a positive charge on nitrogen, it interacts with all these oxygens, lone pair of electrons on oxygen. But if you remove this proton from the ammonium, dibenzyl ammonium salt, then what will happen? It will become simple amine, and amine has lone pair of electron on the nitrogen, and therefore it will get repelled by this because they also have lone pair of electron. So if you can remove this proton, using base, it will come out and it will no more be pseudo-rotagens. If you will again protonate this, it will go inside and it will stay there for some time. So you can do this switching by using base and acid, and therefore, it has some properties of molecular machine. We compare our synthesized compounds with this reported compound, and we observe that this reported compound, with this dibenzyl ammonium salt, have very less association constant, 320. However, our synthesized compounds has very high association constant, 480. This is 689, almost double. So why this high association constant? It's very easy to understand, because in the synthesis, in the reported compound, there is a aromatic ring here, and this aromatic ring removes some of the electron density from these oxygens, and therefore the interaction is less. However, in our case, this oxygen is not attached to, none of the oxygen is attached to any aromatic ring, and therefore it is available for interaction with this positively charged nitrogen, and therefore the association constant is very high. So I will finish my talk here. What I wanted to tell you is that lipase mediated diastereoselective deacylation and synthesis of conformationally restricted bicyclic nucleosides, which can be used for synthesis of modified oligonucleotides. And we have also done, this micelle synthesis, which can be used for drug delivery application. And at the end, I have told you that the same sugar we have used for synthesis of this molecular machine kinds of things. So this is what we have done recently, and I hope I have conveyed you the message that how the chemistry is the center stage of natural science. These are my students who has helped me in carrying out this work in the lab. It is because of them I could tell what I have told you today. It is their hard work, and these are some of the funding agencies which has helped us, because to do the research you need some money. And here are some of my collaborators, Professor B.S. Parmar. We have started lipase-mediated organic reactions together long ago. Professor Rainer Haag, who have helped me in this encapsulation study, and this Professor Sunil K. Sharma, who has used in characterization of these polymers what we have synthesized. So they are my collaborators, and I am very much thankful to college and to all faculty members of chemistry department and principal of the college to give me this opportunity, to tell you some of my work and put my work in the context of, overall work going on globally in the area. Thank you very much. Thank you. So now I will pass the mic to Dr. Arora.
Thank you so much, sir. That was really a wonderful session, I would say. And
yes, I will be very happy to take up if there is any question.
Yeah. Okay. So when I first got to know the topic of your talk, I was highly intrigued. We all know the importance of chemistry, but the way you've explained such complexities of nature with such ease, that was something of different level. Only a brilliant scientist and a great teacher like you could have done that. This took me back to my days as a student when I first, studied about replication, translation, genes-based pairs, etc. But I would have to say that nobody explained it the way you did with such simplification. I personally gained so much as a student, and also as a teacher, I picked a few nuances from the way of you speak, which I would definitely try to use during my lectures as well. And I am extremely thankful to you, and I'm sure all our listeners are. There are so many compliments pouring in the chat box, and some of the participants wants to share this knowledge you shared today with their students as well. So dear participants, this talk is available on YouTube channel of Shivaji College. You may access it from there, and we'll definitely share the link of the same with you later on through mail. So participants, link of feedback form has been shared in the chat box. You can fill it. And Prasad sir, if you can spare a few more minutes, we can take few questions of the participants.
Sure.
Okay, thank you so much, sir. Sir, there is one question from Dr. Shilpa Jain. How to keep interest going on in pure sciences in the current scenario, means this post-COVID era?
I know that there was a lot of loss of time due to this COVID, but now, particularly for research scholars, the labs are open and they are coming, and we can see that there is a tremendous change in the potential of delivery of product of chemistry, you can say, because even if you take this coronavirus, it is a messenger RNA based virus, and the number of nucleosides is not very large in messenger RNA. It's 106 or 120. So it is a very small strand of messenger RNA, is a small strand of RNA, which enters in the cell, and then it uses cell machinery to synthesize many copies of their own, not many, billions of copies of their own. And then in this RNA strand, then they synthesize particular proteins, which is toxic, and then we get problem. And now you see, once this sequence was studied, and then they synthesized complementary RNA strand. And therefore, all these vaccines immediately came in a couple of months in the market, which used to take 10 years earlier. So because of this pandemic and the need of the hour, the scientists also came together. And in a couple of months, they did what used to be done in 10 years, 15 years. And the vaccine is there in the market. And because of the vaccine, our lives are saved. So many countries, which has 100% vaccination, they said that, we don't have to fear now. But at the same time, we also know that this virus is getting mutated, changed to another virus. And why this change happens? Because these viruses are dividing very fast. One day, billions of new viruses appear. So every division requires to form new virus, means new RNA, and nature does mistake. So when they are synthesized, some oligonucleotide get changed in the strand, and then it's a new virus. So it's a big race between humans and the virus to find out how to control this. They can mutate just because it's a natural process of mutation. But then when they mutate, some of the mutants can be very dangerous for us. So it's a big race going on, and we know what is the result. Of course, we are very positive, and things are coming in a positive end. We had a very tough time a couple of months ago, but now the same Delhi, you see, there is no cases. Newspaper says nil, or 5 or 10 or 20, and those were days. It was in lakhs. So it's a race, and certainly science is delivering here. Many of these vaccines are RNA-based vaccines, except few. For example, our Covaxin is a virus-based vaccine. It's not RNA-based. So there are only few which is virus-based vaccine. Mostly it is RNA-based vaccine, because we know what is the sequence of RNA of the virus, and then we can prepare complementary strand, and we can block them, and then their expression will stop. Or else we can select only that portion of the virus which synthesize this toxic protein. So we will take that strand and we will put little bit of that strand in the biological system in our body. So that protein will be synthesized, although we don't have corona infection, but we have that little bit of strand, and that strand will synthesize that protein, and then our body will think that it is the virus, and then our immune system will develop. So we will not be killed, but we will develop our immune system. That's what these vaccines are doing. So this is a good challenge which has been thrown during this pandemic period, and it has been taken care of. So this is what has happened during this time.
Thank you so much, sir. So next question we have from Khushi Indora, and her question is, sir, is CRISPR technology used today in hospitals to treat genetic diseases?
It is in the developmental step, but I could see that more than 10 companies has come which is using CRISPR technology to bring drug in the market. I came to know that even one drug has already come, because that once you know the sequence, it is very easy to develop a drug because the sequence is known. And this sequence, when you work on DNA and RNA, it is always based on Watson and Crick hydrogen bonding, and therefore a specificity is very high. So because a specificity is high, it will not go and stick to another place in the DNA. It will go only in that region because it has a specificity complementary to that region, and therefore it will go only there. However, all these small molecule drugs, it goes to a particular protein which is disease protein, but then a specificity is less, and therefore it goes and work on another protein also, and therefore it causes side effects. So therefore, a lot of these side effects can be taken care of by working and developing slowly medicines based on this CRISPR technology or this oligonucleotide business. And a lot of company has already started. Even one of my students who is in US now, he is also working for a company which is working on CRISPR technology. One company's name is CRISPR and something, So this is really coming up in a very fast way. But everything, nature is great. Nature, how the nature will take to this task, it is difficult to say. Because whatever the small molecules we are using today, they have been not used today. They are using from thousands of years. That has come from our traditional knowledge, and then we isolated compound from plants, for example, artemisinin. So they have come somehow from our traditional knowledge, and those things are being used from thousands of years ago. But this technology has come now. How effective it will prove in future, it is very difficult to say. But scientifically, you can say that it's very accurate and very specific.
Thank you so much, sir. So next question we have from Dr. Mir Mohammad Masood. And the question is, sir, let me know whether any institution in our country research on gene editing is taking place.
Recruitment.
Yes, sir. Any institution which is wherein the gene editing research on this field, gene editing field is taking place.
There are many labs. I'm sure in India also many people will be working on this CRISPR technology. So there are many. I cannot tell you specifically, but my student is working in UMass Lowell, near Boston, and there the whole company is working on this CRISPR technology. Certainly in India also there may be many research groups. I don't know if any company has come up in India, but there are many research groups who are working. And even in US, if they are working CRISPR technology, there are many companies in India to whom they ask to synthesize the oligonucleotide. They will give the sequence and they will ask, please deliver me this sequence. So they may not be working directly, but they're working on synthesis of oligonucleotides since many years. So many companies in Hyderabad, in Mumbai, they are expert in synthesizing modified oligonucleotides or simple oligonucleotides. Even you can buy. I mean, buying normal oligonucleotide is very easy. You just tell them, and it takes per nucleoside some couple of thousand rupees. So if it is 20 nucleoside oligonucleotide, so let us say 2,000 into 20. So it's not very expensive also. But when it is modified nucleoside, then it's expensive. So many companies are working on this in India also.
Thanks a lot, sir, for clearly answering the queries of the participants. And we can see in the chat box, it's kind of an interaction like we have in our class. So we all have become students today. So they are, still, giving you compliments. They are actually, I guess we all are blown away by the simplicity you have explained, such, complex phenomena with us today. So thank you so much for that. I now request the senior most faculty of Chemistry Department of Shivaji College, Professor Manjula Singh, to give official vote of thanks. Over to you, ma'am.
Thank you, Richa. Thank you very much. And welcome to you, sir. I have been longing to see you like this.
Thank you.
And first of all, thanks to Almighty God for showering blessings and grace, and may it always prevail over all of us. It is my privilege to propose a vote of thanks on this occasion. On behalf of the college and the department, I express my sincere thanks and gratitude to you, sir. And it was a very enlightening and enthralling experience, this lecture of yours. I am very thankful to you for all this, and I'm very thankful for your contribution to Shivaji College. As a chairman and member of the governing body Shivaji College, I have witnessed your unparalleled wisdom in the decisions made for the welfare of the college. You are a big source of inspiration and strength. Words are not enough to thank you. We are very fortunate to have you. I've seen an exhibition of your amazing wisdom and the policies you make. So we are very thankful to you, sir. I thank Professor Shiv Kumar Sahdev, principal, the backbone of the college, for his always support and help. I thank Professor Darshan Malik, coordinator of Star College Scheme of Shivaji College, for her always smiling and supporting nature. I extend my thanks to the star workers of this program, Dr. P.K. Sahu, convener, and Dr. Richa Arora, co-convener. And finally, all the faculty members of the Department of Shivaji College, Chemistry Department, for putting their heart and head in organizing this event to perfection. My thanks are due to all the lovely participants for their enthusiastic participation. Lastly, I wish you all the best of everything in life. God bless you. Thank you.
Thank you very much. Thank you, madam. Thank you.
Thanks a lot. Thanks a lot, ma'am. And once again, sir, I would like to thank you for delivering such a wonderful talk with us, with all the participants. We have teachers over here, we have students over here from across the country. So on behalf of all of them, I thank you. And I also thank all the participants for joining us today. Sir, with your permission, I would like to formally close the webinar.
Thank you. Thank you very much. Thank you.
Thank you, sir. Thank you.
Thank you, everyone. We hope to see you again next time, and have a good day.